associate professor at Department of Biotechnology in MNNIT, Allahabad, Prayagraj. Now Prayagraj. So he did his PhD from Center of Bioinformatics, University of Allahabad, and he earlier his post graduation was also from by University of Allahabad. He has vast teaching experience since 2012. He has been associated with Motilal Nehru National Institute of Technology, Allahabad. He has supervised many sponsored projects. The sponsoring agencies were Served New Delhi, CST-UP, TIPQUIP, MHRD. He has been a PhD supervisor of more than 6 students. Several MTech thesis supervision he has done. He has been supervising BTech thesis as well. He has published several papers amounting to hundreds of papers, research papers in reputed journals. Now we have with us Dr. Ashutosh Mani, who will be presenting his research work on machine learning based identification of diagnostic markers for colorectal cancer. So now I welcome you, sir, on behalf of organizing committee, and I would like you to start with your presentation so that our participants can enrich from your lecture.
Am I audible?
Yes, sir, you are quite audible.
And my screen is visible to you?
Screen is visible.
Okay, okay. So first of all, I would like to thank Dr. Nidhi Mishra and her team for organizing this very relevant course, and also for providing me this great opportunity to interact with you all. So I'm also thankful to IIIT Allahabad, and I would like to present my work, that is ML-based identification of diagnostic markers for colorectal cancer from RNA sequencing data. So my screen is visible. The slides are properly visible to you.
Yes, sir, it is visible.
Right, right. Okay. So that's great. I think we can proceed ahead. Right. So we all know that cancer is a very big concern for researchers and scientists, because it is expanding at a very in a very large population, and many risk factors are involved associated with occurrence of cancer, and especially colorectal cancer is supposed to have a lot of factors involved along with it. It may have genetic as well as environmental factors. But uh, today's talk that focuses on colorectal cancer. So we are just trying to identify some markers, so that can act as diagnostic markers, because early diagnosis of cancer can help a lot in fighting with the disease. And there are some markers for identification of cancer, but still we don't have sufficient markers that could classify different types of cancers, like colorectal cancer or lung cancer or ovarian cancer. So people or scientists across the globe, they are working to identify some specific markers, and many markers have been identified, but still there is a lot of confusion that this marker is also present in this marker is also present in this. So ambiguity is there, right? And with the introduction of AI and ML tools, because we are dealing with sequences or reads or data in genomics, and we are getting data from next generation sequencing experiments, and it can be handled with the help of AI and ML also, analyzed with the help of AI and ML also. That's why there is a lot of focus on using AI or especially machine learning based tools to analyze genomic data, and especially next generation sequencing data, because it is data is very huge, it is big data, and analysis with usual tools or manual methods is not possible. And machine learning based method tools are quite helpful in classifying data that is coming from diseased individual, or whether it is coming from normal or healthy individual. So such type of classifications can be easily done by machine learning based methods. That's why a lot of focus is there on machine learning. So now we come to colorectal cancer again. So basically it is a malignancy, and that affects the colon or rectum as well, and both cases may be there. And these both are they are components of human intestine. And the as I told you that it is very much prevalent, especially in individual who are above 40. So they are at high risk, and the data suggests that approximately 1.1 million new cases each year they are reported. And notably, colorectal cancer is the second most contributor to cancer induced mortality. So it is common also, and the problem is that it is detected very late, though some symptoms are there, but people don't pay so much attention, because the symptoms are quite similar to other ailments that are not so much dangerous, and they generally may not be identified without proper medical intervention. And recently I studied one interview. I just came through one interview that was I think Suhasini Ali was there. So she has been recently diagnosed with the stage 4 colon cancer, possibly matlab, if I'm not missing it. So 5 or 10 days back I just learned it. So she said that I was regularly in touch with doctors, but they misdiagnosed. They could not correlate that it may be cancer. So it may be missed, and it may come to stage 4 or and so on. So the problem is that we don't have exact markers that could identify that it is cancer or not, it is colon cancer or not. So that's why machine learning is being introduced. And uh, the statistical estimations are that by year 2040 there could be 3.2 million new cases of colorectal cancer worldwide, and that would lead to 1.6 million fatalities. So that's a big number, and we have to become very much prepared to combat it. And another agency is there, that is Globocon, that it has predicted that particularly in India, the colorectal cancer cases would increase to 2.08 million. right? So it it shall contribute to rise up to 57.5% individuals from 2020 to 2040. So it's a big jump in case of colorectal cancer individuals. And food and dietary habits, other environmental factors, as well as genetic factors are contributing a lot. So this is a comparison of gender-wise comparison of estimated new cases and estimated deaths that are caused by different types of cancer, and colon cancer cases have been highlighted in the red. So these are data of 10 leading cancer types, right? So it is taken from a study published in 2023. So we can see here that 8% deaths are caused by colon and rectal cancer in males and females. Sorry, 8% new cases are there due to colon cancer in males and females, while deaths is a number of deaths is greater in males due to colorectal cancer, that is 9% in comparison to females, that is 8%. Right. So this is again a comparison of I think I can enlarge the table so it may be more visible, right? Okay. So again, this is comparison of gender-wise comparison of cancer incidence rates, food incidence rates, and age-adjusted rates for occurrence of cancer. And the data suggests that for colon cancer, the chances of having colon cancer in males is 1 in 260, and in females it is 1 in 298. And for rectal cancer, the chances of occurrence of rectal cancer in males is 1 in 200. How much it is? Yes, 244, and it is 1 in 295 in females, right? So it is just data that suggests that how colon cancer is one of the main causes of death worldwide, among the other deaths caused by other types of cancer. Right. Now we come to the reason or risk factor reasons, or genetic factors, or metabolic pathways that contribute to colorectal cancer or other types of cancer. And we can see here that there are different pathways that have been mentioned in this slide that represent different pathways that are involved in colorectal cancer. So some pathways are, many pathways are common in different types of cancers, and there may be variation or contribution of, different pathways may vary depending on the cancer or other genetic factors. But these pathways are responsible for occurrence of cancers in individuals, right? So we can just talk about some pathways like WNT catenin signaling pathway, or there is EGFR pathway, or we can talk about EGFR, or that is also called MAPK pathway. And the pathways AKT pathway or AKT signaling pathway, or there is not signaling pathway, and they all somehow they all contribute to occurrence of development of cancer. And it may be colon cancer, it may be other type of cancer, but we are just focusing on colon cancer in this case. And we can see here, right? And different proteins or different mutations may contribute additionally, that can trigger a pathway that can be responsible for generation of cancerous cells, right? And that's so that's about pathways. And there are 4 main grades of colorectal cancer. So there are, yes, it is visible. So there are 4 main grades of colorectal cancer. So the it is shown here, right? So we can see here that the first stage is defined as mucosa or lining of the colon or rectum, while the organ wall has not yet developed the tumors. Here we can see here the organ wall has not yet developed the tumor, so mucosa or the internal lining or epithelium is quite proper, right? It is in well shaped and in order, right? And after that, there is second stage where the walls of colon or rectum begin to develop tumors, and the neighboring limbs or nodes are not affected at all. So there is the position or cells that are starting to develop tumors, but the neighboring cells of the epithelium have not been affected at all. In the next stage, the polyps start forming, right? So here we can see that neighboring cells have been affected. They are also developing cancerous cells, and the polyps have started to form. And in the next stage, that is also part of the second stage, the adenoma has formed, right? So it can start the formation of bulged epithelial cells, a group of epithelial cells that is formed, that causes formation of adenoma. And in the last stage, carcinoma has developed. We can see that the proper bulging is there, and the tumor is there, and it is it has formed carcinoma. And at the last stage, in the last, the tumor has become invasive. So it has crossed the epithelial line, and it is now expanding, or it is affecting the internal lines across the epithelium, or the tissues across the epithelium, right? Internal intestinal wall is being affected, right? And as a result, the disease, or the cancerous cells or the cancer is metastasized, right? So these are the main stages of colorectal cancer. And Now we come to growth signaling and different pathways again. Uh, as I told you, there are several pathways: not signaling, MAPK, WNT signaling. So they all are affecting the they may contribute to development of cancer in healthy cells, right? And the I can enlarge it, I think. Yes, is it visible? Can you see it? Hello, can you see it?
Yes, sir. It is
Yeah,
visible.
yeah. Okay.
Yes, sir. Yes, sir.
Yeah. So now it is more clear. Here we can see starting from VEGF signaling and RTK pathway. So here we can see that it is inducing angiogenesis. Angiogenesis is a very peculiar feature of cancerous cells. Here we can see that uh, CTLA or PARP inhibitors, they are causing genomic instability and mutations. There are telomerase inhibitors. They are calling telomerase dysfunction. There is EGFR signaling that I just told you. It is sustaining the proliferative signaling, right. And there are CDK inhibitors. They are evading the growth suppressors. So despite of there are despite of these uh, cancerous cells, they are dividing at very high rate, but still they are being evaded by suppressors, right? And there are radiation or trail activators. They are resting the cell death. So despite of being cancerous, the cell is not dying. Okay. There are IDO inhibitors. They are deregulating the cellular energetics. Okay. And I started from here. And there is TGF beta inhibitors. They are causing the activation of invasion and metastasis. So they are causing the cancer to spread, right? And so these are so we can target the cancerous cells by different therapies depending on the molecule that is involved in proliferation or development of cancer. So different therapies can target different molecules according to their role in development of cancer. So this is just an illustrative image that shows that how we are devising different methods that are targeting the hallmarks of cancer. Right. So now we come to machine learning based methods. So machine learning is right. So why what is machine learning, or what is artificial intelligence, or how artificial intelligence is a larger domain, and machine learning is a type of AI. In machine learning, we are training a deep machine to learn on the basis of experiences, or maybe it may be learning may be supervised or unsupervised learning, but it is based on experiences. So we are trying to learn in the same way as we are trying to make machine learn in the same way as we are learning. We are also learning from experiences. So primary purpose of machine learning, algorithms, primary purpose of machine learning algorithms is to extract features. So features are properties, and uh, in
कितना अच्छा
DNA.
समझा रहे बेटा.
Hello. Yes. Hello. हाँ. हेलो. सो हेलो. किसी ने कुछ कहा क्या? अच्छा. नो. सो machine learning basically it tries to extract features. So features are properties. So in DNA, the properties are in DNA or genomic data, the property is order of sequences or order of nucleotides. If we are using nucleotide sequences like DNA, then or RNA, the order is nucleotide, right? The nucleotide bases that are this is the order. So when we are considering NGS data or genomic data, so we are interested in features. So these features are the feature is the order, the property is the order of nucleotide. So generally, in machine learning algorithm, we are taking features from the structured data, right? So we have structured data, and the what we do, we just feed the we fed the input in to the machine learning algorithm, and uh, we get the outcome. We get some a prediction or some result. So we get the outcome, and in this outcome may be in form of some classification or some prediction, and like in case of colorectal cancer, or the data that we are getting from sequencing experiments, we are just interested to know that whether this marker is is able to, or this gene is able to predict accurately or not that there is cancer or not. So we are interested to know that there is a gene and its sequence, and its expression level. So we all have same genome, we all have same set of genes, but their expression levels are different. So in RNA sequencing, we are sequencing the expressed RNA, and the number of genes are same in each individual, but expression of each gene may vary according to certain condition. So suppose there is gene 1 and gene 2. In normal condition, gene 1 expresses 10,000 copies, gene 2 expresses 10,000 copies. So both genes are expressing 10,000 copies for one protein expression. But what happens, suppose in case of colon cancer, gene 1 start forming less copies, like 5,000 copies, and gene 2 starts forming 20,000 copies. It has just double. Gene 1 has reduced to half, and gene 2 has just double. It means that the expression levels are changing for these two genes. For gene 1, in case of colon cancer, expression level becomes half, and in gene 2, in case of colon cancer, expression level becomes double, and is right. And this, if this can be directly correlated with the occurrence of colon So that's very important, very significant contribution. And the things are not so much easy when we are talking about genomic data or RNA sequencing data. The data is very complex. The data is very huge, and we want to that's why we are interested in machine learning based approaches or algorithms, because or computational method, because uh, expression levels are highly varying, number of genes is very high, reads and we have to adjust a lot of a lot statistical parameters. After only that we can say that this gene has expressed so much, and even after that correlating these expression levels with certain type of cancer, or and further classifying it, right, that this gene has expressed so much in comparison to normal, then it's colon cancer or it's not colon cancer. So that's a complex process. That's why machine learning methods have become so much useful. So as I told you that there are two major categories of machine learning methods. One is supervised learning, and one is unsupervised learning. So it is clear from their names that in supervised learning we are just supervising, we are providing the previous feedbacks, we have the training data sets that have been fairly classified, that colon cancer, non-colon cancer, or cancer or non-cancer. But in unsupervised learning, it will classify automatically, right? So we have to provide in supervised learning. We are generally providing features. We are yeah. So so it may be it may dependent it may be depending on your requirement, that which type of algorithm shall be used for predicting or classifying this type of data sets, or diseases. It depends on your individual requirement, according to the disease that has been selected, the type of data that has been selected, and so we can do further analysis with the help of machine learning methods. So there are so it's okay. I think we have talked about machine learning and its introduction. So there are methods, various methods like support vector machines, or random forest methods, or neural networks, or that are artificial neural networks, regression models like linear regression, or ADA boost, or Gaussian biases. So a lot of methods are there, but and we can test and try different methods for prediction of accuracy, prediction with accuracy and their sensitivity. A lot of exercises can be done, and the ML method that best suits, or that is able to classify with highest accuracy, may be opted further for a classification of the data or predicting the disease. It depends. It depends on the results. So actually, I am interested in discussing the workflows that have been recently used by different researchers for dealing with RNA sequencing data and identifying the potential biomarkers. And so this is the first workflow that we have used. So let me from the audit, right. So here we can see that we have taken the colorectal cancer data from TCGA, right? So here TCGA CRC data set was taken, and we first, identified the, differentially expressed genes. So in one approach, we we found the read counts, and we identified the differentially expressed genes, and we identified the significant disease. And in another method, we applied ML based approach to identify these genes. So what we have done, we applied ML based approach. We performed class balance and feature selection. We just balanced the classes and selected features depending on the data, and after that we perform the ML classification depending on the selected features. So what we found that by using both methods, we found different sets. You're right. So by using both methods, we found different sets. But interestingly, there were 38 genes that were common, right? And we were sure that by both methods, the genes that are common must be significant. They must have high correlation with the occurrence of cancer, because their differential expression is causing colorectal cancer, right? So we further performed survival analysis and expression analysis, and on the basis of correlating it with the literature and biologically validated information, we identified a set of genes, and the most important gene was TMEM236. So we propose that this is a potential novel diagnostic marker for colorectal cancer. So the study was published in Scientific Reports in 2021. So we can see here that the method, the workflow, the standard workflow for differential expression of genes, and the ML-based workflow for identifying differentially expressed genes had 38 genes had shown that there were 38 genes that are commonly differentially expressed, and their differential expression was significant, right? And so we are in this method we applied dual approach. It means that same read counts, same data were analyzed by both methods, by our bioconductor pipeline as well as ML approach, right? And as a result, we were able to identify a set of genes that are commonly expressed, and it is clear that if the both methods are identifying the same set of genes, then the expression or expression level or and as well as genes both are important, and they have high correlation with the occurrence of colorectal cancer in patients. And the next is second workflow. So it was another study where we had taken the TCGA data set in one experiment, and parallelly we had taken CRC gene expression omnibus data sets, and in the first experiment or first parallel experiment, we identified DEGs by TCGA biolinks, and in the second parallel experiment we identified the DEGs or significant DEGs by DSEC2 and top contact. So both these are tools for identification of differentially expressed genes. And again, so again, interestingly, we found 170 genes that can be, said to that can be directly correlated with colorectal cancer, and they were common in both, results. So they were the important thing is that the data sets were different, the experiments were different, right? And both were processed through two different approaches. In first approach we used TCGA link, and in the second approach we used DSEC2 workflow, and still we found that there are 170 genes that are significantly differentially expressed in colorectal cancer data. Okay, and both data sets were totally different. The methods were also totally different, but 170 genes are common. It simply means that these 170 genes are really very important, and their differential expressions are very important for occurrence of cancer. So further, we applied a Baruta algorithm and identified features or selected features from the TCGA and CRC data sets with 170 genes, and after that we found that there were 43 confirmed features, and we rejected 94 features, and there were 33 confirmed features and 43 tentative features, and the tentative or rest of the features were rejected, or they were not included in further steps, and we just focused on 33 confirmed features. And by using these features, we applied random forest method for prognostic classification of the model, and further we cross-verified the outcomes with overall survival analysis, and we found that when we correlated the genes and immunocytes, we found that there are two genes, VSTM2A and GLP2R. There were two genes that were significant, they were very relevant, they were able to classify CRC data and non-CRC data with highest accuracy. And we proposed them as a novel colorectal cancer biomarkers, right? So this study was again published in scientific reports in year 2023. So this is the a workflow that we had used for construction of colorectal cancer prognostic model development and further screening of prognostic risk genes using machine learning algorithm. So here we have GSE 126092, GSE 156355, and TCGA. So two GSE data and one TCGA data, and after that, we found 330 differentially expressed genes, and by using survival package, we found that there were 40 genes that after unvariate Cox regression and lasso regression resulted in 17 genes, and survival analysis followed by prognostic risk, models, application of prognostic risk models, we were able to identify the set of genes that were, correlating colorectal cancer uh, by accurately classifying the disease on the basis of RNA sequencing data. And again, this is a prognostic model based on immune-related genes in colorectal adenocarcinoma. Right. So we can see here acquisition of code-related genes, colorectal adenocarcinoma-related genes, data from TCGA, and that were imported clustron project. And further, we found differentially expressed analysis. We performed differential expression analysis, and only significant changes were uh, included, and those were having statistically insignificant changes were excluded from the studies, right? So further, this is the workflow where we propose for unlocking the potential of CA2, CA7, and ITM2C gene signatures for early detection of colorectal cancer. So it was a comprehensive analysis for RNA sequencing data by using a machine learning algorithm. So here in this case, we had taken TCGA and CRC datasets, and CRC datasets, and we performed feature selection, and after that we used 5 different algorithms, right? AdaBoost, random forest, linear regression, Gaussian naive Bayes, and support vector machines. And we found that random forest was best performers. Its predictions were accurate, more had shown highest accuracy, and we found that there were 314 features that were significant. They were highly relevant, important from the TCGA CRC datasets, and 503 features from GSE datasets. And both these datasets had 12 common genes. Both these datasets had 12 common genes, and we performed gene expression and correlation analysis, and we found that there are 4 genes that can be helpful in early detection of colorectal cancer by because they can be used as biomarkers, diagnostic biomarkers for identification of colorectal cancer in patients. So these were the genes, and finally we prepared a set of genes that can act as biomarker for prediction of colorectal cancer by using RNA sequencing data. And that's all. Thank you.
So, dear participants, if you have any queries regarding this lecture, you can ask sir directly or write in the chat box. So, participants, I think you don't have. Okay, so it is lovely that who has asked this question. What is specific machine learning model for developing the prognostic classification model?
That's a nice question, and there is no specific model. As you can see that in one model, in one paper we found that random forest is working very well. In another method we found that support vector machine is working very well. So it it depends on the data or the features, or what is type of classification that that you want to classify. So you have to check all possible algorithm. We have to apply all possible algorithms for classification, and only after that you can come to know about the accuracy with which they are predicting or classifying the data. And the best the model or algorithm with best accuracy can